Bismillah ar-Rahman ar-Rahim. Today we are going to discuss an important lecture about the metabolism of red blood cells. Red blood cells main functions is to carry oxygen and deliver it to the tissues, and on the same time to carry carbon dioxide from the tissues and bring it back to the lungs. So the main function is to carry oxygen from the lungs to the tissues and carry carbon dioxide from the tissues back to the lung. For that reason, the red blood cells are not having organelles. We can resemble the red blood cells as bags of hemoglobin. Most of the contents of the red blood cells is hemoglobin. No organelles and no nucleus inside the red blood cells. That would mean that no mitochondria, so there is no respiratory chain. There is no respiratory chain, so ATP obtained only by glycolysis by substrate level phosphorylation. Again, RBCs are not able to carry out fatty acid oxidation. There is no gluconeogenesis in the red blood cells. NADH, when produced during the process of glycolysis, cannot enter into the respiratory chain to be oxidized. So how NADH is oxidized inside the RBCs? It is oxidized by reduction of pyruvate into lactate. For that reason, the lactate is the end product of glycolysis in RBCs. So we have to observe that red blood cells are not having mitochondria, so all the ATP obtained by the red blood cells is obtained by the process of glycolysis, and glycolysis will proceed anaerobically. As we all know that glucose is taken by facilitated diffusion by specific glucose transporters present on the cell membrane of red blood cells. They are termed GLUT1. Such glucose transporter is insulin independent. Its level on the surface of the red blood cells is not dependent on the presence or absence of insulin. Now, this is the process of glycolysis which we had studied before. We can notice that the process of glycolysis, as we see glucose, then glucose 6-phosphate, etc. But the most important is that when NADH is produced, it is reoxidized again by the process of conversion of pyruvate into lactate. So the only process for oxidation of the reduced coenzyme 1 or NADH is by the conversion of lactate into the conversion of pyruvate into lactate. There is no mitochondria, no respiratory chain, so there is no reoxidation through the respiratory chain in red blood cells. For that reason, again, hemolyzed blood sample will show elevated levels of lactate dehydrogenase, because lactate dehydrogenase is present in excess inside the red blood cells, which when destroyed or hemolyzed, it will leak into the blood plasma and its level is increased. For glucose estimation, as we mentioned that before, we are adding fluoride to the blood samples. Why? To inhibit the process of glycolysis, so red blood cells will not utilize glucose and decrease its level, which may give false low results of glucose. We add fluoride because fluoride inhibits the enzyme enolase, so it will inhibit the process of glycolysis. Let us see again another observation on the process of glycolysis in RBCs by noticing that pyruvate kinase deficiency will cause hemolysis. Why pyruvate kinase deficiency will cause hemolysis? Because pyruvate kinase is a step at which substrate level phosphorylation of ADP into ATP is produced. So the source of ATP synthesis is a pyruvate kinase. If pyruvate kinase is deficient, there is no enough quantities of ATP inside the RBCs, so the red blood cells cannot maintain its integrity. The cell membrane of the red blood cells cannot maintain its integrity, and the red blood cells will hemolyze. This is one of the enzyme deficiencies that when inherited, it may cause hemolytic anemia. Another pathway in the process of glycolysis in the red blood cells is a side way from the conversion of 1,3-bisphosphoglycerate into 3-phosphoglycerate. Such a side pathway, we call it RL cycle. By the RL cycle, 1,3-bisphosphoglycerate is converted to a very important compound or molecule for the red blood cells to function. This compound is 2,3-bisphosphoglycerate or 2,3-BBG. 2,3-BBG generated from 1,3-BBG by an enzyme mutase. So mutase will convert 1,3-bisphosphoglycerate into 2,3-bisphosphoglycerate. 2,3-bisphosphoglycerate is essential for the function of hemoglobin because 2,3-BBG decreases the oxygen affinity of hemoglobin. The presence of 2,3-BBG makes hemoglobin molecule more deliverable for what it carries from oxygen to the tissues. If 2,3-BBG becomes deficient, as in stored blood, the hemoglobin affinity toward the oxygen will become high, so hemoglobin will not deliver oxygen to the peripheral tissues. 2,3-BBG is then converted into 3-phosphoglycerate by means of a phosphatase enzyme present inside the red blood cells. So a side way from the process of glycolysis by which 2,3-BBG is generated, and 2,3-BBG, because it is a negatively charged molecule, it lies inside a positively charged molecule, resides between the two beta subunits of the normal human hemoglobin, makes the hemoglobin molecule tetramer more readily delivered of oxygen, and makes its affinity toward the oxygen less than in its absence. So 2,3-BBG will deviate or shift the oxygen saturation curve to the right. In case of stored blood, glucose inside the red blood cells will be consumed, and as a result 2,3-BBG levels will become less. Lowering of the level of 2,3-BBG will increase the oxygen affinity or the hemoglobin affinity toward the oxygen. So stored blood will not deliver properly what it carries from oxygen, and this is serious for the patients or who suffer from anoxia. So stored blood has lower concentration of 2,3-ppg. To solve such a problem, inosine is induced to the stored blood. Inosine, because it is uncharged, will pass through the cell membrane easily, and inside the red blood cells will be converted back into 2,3-ppg, restoring the normal level of 2,3-ppg inside the red blood cells. The question is why we cannot add 2,3-ppg to the stored blood. 2,3-ppg will not pass the cell membrane, being a charged molecule. It is hydrophilic, so it cannot pass the cell membrane of the red blood cells. Now we are going to shift about antioxidants and stability of red blood cells. What are the importance of antioxidants and the stability of red blood cells? Antioxidants play an important role in protecting red cell membrane against oxidative stress, and consequently they protect the red blood cells against hemolysis. Certain drugs or food can lead to generation of reactive oxygen species, or we call them oxidants. They can produce hydrogen peroxide. The hydrogen peroxide generated can carry a chain reaction that free radicals react with other normal compounds, converting them into free radicals. So once free radicals are generated, they will carry out a chain of reaction that can produce more free radicals, and such free radicals will cause cell destruction. Antioxidants, example, tocopherols, vitamin C, or beta carotenes scavenge such free radicals and protect against hemolysis. There is certain enzymes and other compounds other than the vitamins as antioxidants that can protect the cell membrane against hemolysis. For example, glutathione. What is glutathione? Glutathione is a tripeptide. Tripeptide means it is formed from 3 amino acids. These amino acids are glutamic, cysteine, and glycine. What's the active group of glutathione? It is the sulfhydryl group, or SH, of cysteine. So glutathione is gamma glutamyl cysteinyl glycine. So how glutathione functions in the process of elimination of free radicals from the red blood cells and protecting the red blood cells from hemolysis. Let us look for the enzyme glutathione peroxidase. The enzyme glutathione peroxidase converts hydrogen peroxide, H2O2, into molecule of water. Water is not oxidant. So the glutathione peroxidase will convert hydrogen peroxide into water. By means of what, or by the help of what? By help of glutathione in the reduced form. But in such a process, glutathione will be converted into the oxidized form, which is GSSG. So glutathione in the reduced form help glutathione peroxidase in the elimination of oxidation or oxidizing agent or a free radical H2O2 by converting it into water, but in the same time glutathione, the reduced form, will become oxidized. To convert the oxidized form of glutathione back to the reduced form, this occurs under the effect of the enzyme glutathione reductase. Glutathione reductase gets its 2 hydrogens to reduce the oxidized glutathione back to the reduced one by help of the reduced coenzyme 2, which is NADPH. In such a process, NADPH will be oxidized into NADP. NADPH is converted into NADP. Again, NADP should be reduced to the reduced form to help the glutathione reductase. This is carried out by the glucose 6-phosphate dehydrogenase enzyme. So glucose 6-phosphate dehydrogenase converts NADP into NADPH by the two reduction or the two steps in the oxidative stage of the PPP or the pentose phosphate pathway. And the first one, or the first step, the step of glucose 6-phosphate dehydrogenase, in which glucose 6-phosphate is converted into 6-phosphogluconate, gluconolactone. Glucose 6-phosphate is converted to 6-phosphogluconolactone. Another dehydrogenation step in which 6-phosphogluconate is converted into ribulose 5-phosphate. By such step, NADP is converted into NADPH. So, what is the source of reduced coenzyme 2 or reduced NADPH needed for the enzyme glutathione reductase? The source of the HMP pathway. By means of which enzyme? By means of the glucose 6-phosphate dehydrogenase. If glucose 6-phosphate dehydrogenase becomes deficient, there will be no sources for the reduced coenzyme 2 for glutathione reductase to act, so glutathione will remain in the oxidized form and hydrogen peroxide will not be eliminated from the red blood cells and the hemolysis will occur. There is other enzymes that help in the elimination of oxidant agents inside the red blood cells, like the enzyme catalase, which again can convert hydrogen peroxide into water. Hydrogen peroxide, when it is produced inside the red blood cells, acting as oxidative agent, is very serious for the function of red blood cells. Again, why? Because it will convert the iron of hemoglobin from the ferrous form into the ferric form. Such condition, we call it methemoglobin. It is a methemoglobin. Who converts the methemoglobin back to the normal hemoglobin? It is the reduced coenzyme 2. So the oxidative stage of pentose phosphate pathway is very essential to supply the red blood cells by the reduced coenzyme 2. If glucose 6-phosphate dehydrogenase is deficient or becomes mutated, becomes deficient, which is a hereditary condition, so neither the glutathione reductase will active nor the process of converting methemoglobin into normal hemoglobin will occur. Glucose 6-phosphate dehydrogenase deficiency is an X-linked genetic disease. It results from any of hundreds of mutations. So mutations of the enzyme is many different types. Such mutations will lead to hemolytic anemia or FAFism in most of the cases of glucose 6-phosphate deficiency. There are varying severity of the glucose 6-phosphate deficiency in different patients. So some patients may have the disease very severe, some may have it very mild. So what's methemoglobin? Methemoglobin results from oxidation of hemoglobin iron from ferrous to ferric. Methemoglobin cannot carry oxygen. Methemoglobin will turn the color of the blood into chocolate colored blood. So the conversion of iron, of heme, from the ferrous to the ferric form, we call it the process of methemoglobin formation. Methemoglobin may be inherited or acquired. Acquired due to drugs or certain chemicals. It may be inherited as in hemoglobin M, in which tyrosine replaces proximal histidine in any of the alpha or beta globin chains. Again, it could be inherited due to deficiency of methemoglobin reductase enzyme, which requires NADPH to convert the ferric to the ferrous iron, as we mentioned before. Methemoglobinemia is treated with ascorbic acid and intravenous administration of methylene blue, because methylene blue acts as a reducing agent. This is a case for a patient with methemoglobin, and we are asking you to understand it, then try to answer the question. If you have any question, then contact me, and I will tell you what's the proper answer for this MCQ. Thank you very much.